Good morning. Thanks for being here. We had a little bit of a delay, but now we are ready to get started. So to begin, we have the pleasure to have a short presentation by Guillaume de Treut. He's going to talk about, I know that's stochastic things. Generating transition paths with Markov bridges. Is this version? Thank you, Guillaume.
Okay, so maybe I should put that thing. 5 minutes, right? Okay, so yeah, I'll talk briefly about some recent work generating transition paths with Markov bridges. So just to give you a little bit of context, it's interesting in many different subfields and contexts in order to study transitions from one state to another. For example, here you have an example of protein folding. It's interesting to know how a protein folds from the unfolded state to the folded state. In another context in genetics, here is a simple example of a system with two stable states. So you have bistability. And this is achieved because you have two genes which are controlled by the same operators, but each gene represses the expression of the other one, but there is leaky expression. And so sometimes due to stochastic excursions, you can have one gene which starts to invert the trend and you go over to the next stable state. And so you can have transitions between two stable states with a lot of one protein and the derivative of the other, and then the symmetric state. And it's interesting to know how do you go from one to the other. Yeah, you have other applications. For example, in more applied mathematics, this is a random walk. And so you see like a particle like this, the y-axis is the position at this time, so it makes excursions. And if you can generate paths which start from zero, let's say, and return to it, then you can use a little trick to generate excursions where you see the random walk doesn't cross the half plane, which is below bottom. And so this is why it is interesting to know how to generate those transitions. Okay. To explain to you what's the problem, this is a simple example where you have a 2D potential, and I want to know how you go from A to B. You can see, so in this case, it looks something like this, right? You have a particle starting in a well which is very small, and then it has to go here. But to go here, it has to cross this well, which is like an intermediate state. So you have to go across a couple of energy barriers. So you can simulate this with kinetic Monte Carlo. So this is a master equation for a stochastic jump process. And if you do this by brute force, you have something like this, maybe you would realize that the problem here is that this well is so stable that it's very rare. It's very difficult to escape it and then to reach this well. It would take a lot of time to do it by brute force, and it's not efficient. And this is related to the problem of sampling rare event statistics. So how do you fix that problem? So this is an approach we took, and in which we define, we realize that basically what you're interested in is the probability that you want to be at a certain state at time t, given the initial condition, but you also want to end up at the final condition at time t at the final state. So it's conditioned both to the initial state as usual, but also to the final state. And you can show that this probability satisfies another master equation. If you're familiar with this equation, this is it. But here the transition rates now are time dependent, and they are proportional to the original transition rates from the previous slide. But now there is a time dependent scaling factor which ensures the conditioning to the final state. And so what is the take-home message from this is that this is an exact representation of a conditioned process. So the good thing is you can use the same battery of tools like kinetic Monte Carlo to sample those paths, but you know now that they will end up in the end state which interests you. And yes, so an example, this is the same problem that I showed before, but now I'm using this formalism. So you'll see that all the trajectories, they start from a given well, and as time goes on, they will all end up in the same state at the final time. And all the paths you sample like this they have the same statistics as if I would taken the one from last slide that made it. This statistic is preserved, but it's just more efficient because the simulations are much shorter now than the one that I had to do before. To conclude, so these are my conclusions. If you're interested, we have a preprint. We also have like a Python package to sample this type of transition bridge. And I don't have time to go into it, but we also have an applications to try to understand cell differentiation and how you go from one stem cell to a differentiated cell type. So similar type of studying transitions. And yeah, thank you. Any question? We'll be happy to answer.
Very much, Guillaume. Questions? I have one. I know that many people here work with protein or membranes, and I think that to really break those systems, sometimes it might be hard.
Yeah.
It takes a long time. Do you think this method could be used in these contexts?
I don't know, because like this typically are transitions. Is not an equilibrium process, right? Is a transient dynamic. You go between, you connect two stable equilibrium state maybe or metastable. So what's happening in between is not really at equilibrium, and it doesn't even really have to satisfy detail balance, to be honest. Yeah, I'm not sure. Like it has. I'm not sure finding the equilibrating the system is the right application for that.
Sometimes you can get stuck in a metastable situation, right? And then to escape the situation and go to the true equilibrium.
Yeah.
This seems to be.
Yeah. But you need to know the end state, the target state, right? So if you know the equilibrium, then yes. But otherwise,
I see. Questions?
Just a curiosity, that kind of looks like an SVD or PCA. Is that what you're looking at? Or?
Yeah. So those are like a way to represent. So in this type of plot, each dot is a single cell. Yeah, and they use the sequencing to get the genetic state of the cells, like the gene profile, gene expressions, and then it's a very large dimensional space because you have a lot of genes, right? It's not 3D, it's p dimensions. But in principle, the cell which has similar expression profile, they are closed in that space. But to visualize it, those people use like reduction dimension reduction techniques, and so it's like PCA, but this is called UMAPs, you know. So they have, it's like more clever than PCA, I suppose. But it's still.
So let's thank Guillaume again. So by the way, Guillaume was a last-minute addition to this set of presentations. So I didn't give all the instructions to him on time. And because of this, he did it a little bit longer than usual. But from now on, I think everyone will do like 2 slides, 3 + 2 minutes. Okay.
Who's the first?
So what's the list that we had?
Sorry?
Qual era a lista que a gente tinha? Tinha uma certa ordem lá na lista, né, pra gente manter essa ordem. Pode ser possível? Que a lista tá meio.
Tempo que eu tenho que falar.
3 mais 2.
Tá bom. Primeiro, o meu.
Pode ser o Carlos.
Carlos, né? Tá.
Okay, so now we have remarkable aspects in stochastic thermodynamics and as hyper-accurate bounds in discrete state Markov systems. Thank you, Carlos.
Thanks, Pablo and Danilo for the invitation. There are distinctive fingerprints of non-equilibrium scenario. The break of detailed balancing, positive entropy production, the fluctuation theorems, the efficiency of thermal engines is lower than the Carnot. And in the last years, it arose some interest about the TUR's relations. What I mean, it's basically it states a trade-off between fluctuation and the dissipation. Here is the entropy production. In other words, if you can increase the precision, for any current, of your system, you should increase the dissipation or by choice. Another way, there is the trade-off. The ratio between the variance and the square of the current is always larger than the inverse of the entropy protection. This is for fixed thermodynamic forces. It arose several interests in this, several generalizations. But in this talk, I am talking about, okay, it's always larger or equal. Equal is in the equilibrium regime. If you are in the non-equilibrium scenario, what is the set of currents which closest to match this? This is the idea of appropriate current. You can find more details in this paper. And what is the advantage about talking about appropriate current? It can give you a tighter bound for the system efficiency. Okay, this is the equation for a generic current. It's basically to count the number of transition from state L to M, and this is the weight of each current. Okay, this is for a generic current, a generic microscopic current. Okay, if you take the average for all trajectories, we will obtain the mean current. And if you consider this, you can consider the variance for that. And you can, in a similar way, you can write the expression for the variance. And the idea of appropriate current is to find the set of parameters D and L, the weight D and L and Dm prime L prime, with maximize your average. And if you take this minimum, this is you find this relation for the appropriate currents. In other words, if you wanted to find the set of parameters which closest to match the fluctuation and the dissipation, it should satisfy this relation. Okay. Of course, but now you should find these parameters for master equation systems, which is our goal here. I will not show the details, but you can follow this straightforward approach for Markovian system, and you find the set of parameters. And okay, this is, I would like to exemplify, for example. This is generic currents, These generic currents produce this ratio between fluctuation and the mean average. This is the appropriate currents. We'll find the special set of weights in it. You show it is the set with closest match the appropriation. Okay, this is for this specific system. Okay, another application is, in several cases, including me, we are interested in application of this system, non-equilibrium system, as thermal engines or as engines. Non-equilibrium engines operate out of the equilibrium. Okay, in this case, you can depart from the entropy production form, and you see we have two work sources. This work is given to your system and partially converted in another kind of, for example, chemical work is converted in mechanical work, or vice versa, according to your interest. And this, a measure of the system performance is given by the efficiency. Okay, now, of course, you know that in the case of a thermal engine, this efficiency should be lower than Carnot, of course, it's obvious. In this case, the system operates at equal temperatures. This efficiency is always lower than 1. Okay, the appropriate bounds can give you a tighter bound, can give you a more information about your system performance. Okay, I will show this. This is the appropriate bound, for example, and it's more informative than the one. Because it not only in this system, it not only reproduce, it qualitatively agrees with they are tighter than your real behavior, but it also gives, it follows the same trends about your system. Okay, to summarize, provides the best estimation of the entropy production for the TUR and also bounds for the efficiency. Okay. Thanks.
Thank you very much, Carlos. Good question. So can you apply these things to active systems, for instance? Because it's interesting that, for instance, when you have cluster formation in active matter, the clusters, they have a interface which is really fluctuating. And I think this is quite related with these variants of the current.
I'd say yes, because it's the idea. I don't know how easy it will be, but the idea is for a generic Markovian system, for a master equation system. I'd say yes,
Yeah.
but you should find the weights for the current.
Yeah, it makes sense. For the active systems, if you do a field of entropy productions, you calculate the entropy productions locally. At the interface, you have the largest values. And therefore, you have the largest variance for these
I'd say
currents.
yes. No, I'd say in principle you can, of course. I don't know how easy it will be.
Yes.
But this idea is valid for a generic master equation system.
More questions? So let's, yeah, please.
Just something, when you say that you have an inequality difference in wireless, that's the classical case.
Yes, the classical case, of course, yes, the classical case. No, it's the classical case. This is what I meant. It's valid for fixing drivings.
So let's think on again.
As pessoas, né? Então elas estão sabendo na hora. É bom a gente anotar aqui
Se você quiser.
os próximos. Você mostra para mim a lista aí?
Aqui.
Obrigado. Então, agora,
You're gonna hear some real biological type of work, no equations. And, we did use some equations in between parts of it, but most of it is just intuitive and messing around with data to see how we can figure out a way of understanding the genetic code. You remember the code has 64 kinds of triplets of bases in DNA or RNA and 20 kinds of amino acids. And there is a code to make correspondences between these things. And this is the basis of how cells work, because you have to have memories in DNA and working proteins. And it's the working proteins that make things stay around. And the memories is that what makes us stay as we are for some time. When we change memories, we become a different species or a different variety. And then we are working with this. How did this correspondence between triplets and amino acids came in? These things were discovered in the decade of 1960, 63, 68, all the correspondences were deciphered. But it's already 60 years from then that we are trying to figure out how did it came about. And we are working with this for almost 30 years already. And we have a pretty complete model for that. And we hope it's the right one. The idea we got is that the correspondences were built in arrangements between pairs of the transfer RNAs. There is one transfer RNA in green, the other transfer RNA in red, and they are paired together mainly through the triplets on the top, which are the codons or anticodons. And that's how the code is organized. Nobody had thought about this before, and these things started in 1995. The inspiration that we get is from the ribosome working. The ribosome is a machine that gets transfer RNAs, each transfer RNA carrying an amino acid that corresponds to it. Then the trick, this is already the correspondence made. The transfer RNA with its corresponding amino acid, and then it will decipher what the gene means in terms of proteins that will be the result. This thing is made through a machine, which is the ribosome. The tRNAs get in and out, and when they are coming in, they bring together an amino acid with it. And when they are together, oriented by the sequence of the genetic messenger RNA, when they get together, paired laterally, and then they transfer amino acids one to the other. The idea we got is that how could this start without the machine? To start without the machine, we imagine that these two things got together by themselves, which is this way. Instead of being laterally associated, they are associated through their own means. Then we have to find pairs of complementary triplets that get together on that, and that can make protein synthesis from the tRNAs alone. And then we started working on this. The thing is pretty well resolved. We call it self-referential model, because one tRNA refers to its complement. And the other is that when they make proteins, the protein also has to refer to the producer. These are the producer's proteins, and the product has to refer back to the producer so that they can recognize each other and then they can work together. When these things get settled, we have a code made. I have the results of that on the poster. We can discuss about that. We are here only saying about the mechanism of encoding and how did this come about. We are saying that the one is complement to the other. It means that the identities of them are also the same one referring to the other. When they are equal type of molecules, one complementary with the other, the self-referentiality is kind of undecided because one refers to the other refers to this, and they could be doing the same thing all over. How can you get non-self out of the self? This set is selfing one to the other, and the complements may be different because each one will correspond to a different amino acid. And then they are in a situation forming dimers where the oligomers are coherent one to the other. And I'll leave the question for you. I'm trying to make this mean a singularity, as physicists used to call the Big Bang. And I'm trying to make this an idea for the Big Bang of biology. When we have biology, we have the genes, the proteins, and things going together. And I'm just taking this idea for you to tell me if I'm using the right terminology for that. We can discuss it later. When these things work, they are non-directional. One transfers to the other transfers to the first, and then they may mean a different, the same thing at the beginning, and they will become a different thing after the 20 amino acids are made complementary to the producer of the hormone. We have the next slide. The next slide is when we get the product, identifying one of the producers. The producer is a dimer, one is complementary to the other, but the products will make the correspondence with one of them at a time. Then those two triplets in the producers will find different kinds of products, and each one may make correspondence to one of them. Then we have one dimer, you have two kinds of genetic materials. And we can progress with this to find how the system of correspondences showed up. We have some data that we got from literature that the dimers exist. They can be purified in laboratorial conditions. Only that when this guy did this, they never thought about code. They were thinking about a different thing. And we are taking this as material determination for the idea that we got. When this thing happened, it is a decoherence type of object.
So sorry.
And we are finished. Yeah. And we can discuss the results from this. I think they might be convincing that it's the right model for people. Thank you.
Thank you very much, Romeu, Guimarães. In the interest of time, I think we can leave questions for another time. Thank you. I like that you left your WhatsApp number here so people can contact you. Thank you. So now we have Diego Pereira. He didn't appear. Okay. So now we have then Pedro Paraguassú. Very much, Pedro. Pedro is going to talk about non-Gaussian heat fluctuations in driven overdone systems.
Hello, everyone. As para você, eu talk about non-Gaussian heat fluctuations in drive overdone systems. This is a work that we are doing. I'm doing in collaboration with Treut here in São Paulo. And the idea is the heat formula for overdone systems lead to wrong results. For instance, if I have a free overdamped particle, it will obey this equation, and I use the heat formula, I will have that heat rate, the exchange of energy between the particle and the vat will be zero. And this does not make sense because I have a free particle and the particle is moving and I don't have any potential, any interaction, just the vat there. So you need to have some exchange of energy to have movement. And moreover, if I calculate the distribution of this heat in the overdamped case and take the overdamped limit, I will find the distribution corresponding to a zero heat. That will, in this case, will be the delta. And the solution is simple. We just need to consider the kinetic energy to solve this correspondence and to solve this problem. So the kinetic energy plays an important role in the heat formula for overdamped system. And I'm interested in see these heat fluctuations in drive systems. Drive systems are systems I just applied at first. So I'm doing work on the system, and this work will be a Gaussian variable because it's just external force, so the velocity is Gaussian, and the work is linear proportional to the velocity. I will have a Gaussian distribution, and without the kinetic energy, without considering the kinetic energy, heat would be equal to work. But this is not the case. I have the kinetic energy. So what I wanted to see is this now Gaussian fluctuations do the kinetic energy. Now that I know that the kinetic energy needs to be present in the heat formula. And for studying this, we consider two cases. A passive case, a Brown motion, user Brown motion, and applying an Excel force. And the self-proportion case, active case. So I have this equation of motion. For the passive, I just ignore the active noise. That's given by this expression here, exponential. This usual. Here I have the thermal bath. And I also consider for the passive case a non-isothermal process. And how I you measure this non-Gauss fluctuation? How I see the impact of the kinetic? I do two quantities. The root loss that measures the tails of the distribution, and I know that the excess of the root loss is known for a Gaussian variable. And since I computing the non-Gaussianity through the kinetic energy, I will calculate this quantity, and also I calculate the pullback linear divergence that compared to distribution. So I have this new distribution that we can find dynamics, but it's not here. This work is a work in progress. And use the pullback line distance to compare and see for which variables I will deviate more from the Gaussian behavior. So for the passive case, I have two temperatures along the thermal process, the particle start initially with T0 and finally TF, some temperature. And I see that as I increase the temperature, I deviate from the Gaussian behavior because my root loss is increasing and my pullback linear distance is increasing, also increasing. Here I consider for a different time of the process. This is just the interval that I measured. And for the active case, I just consider the isothermal process because as long as isothermal is not relevant experimentally. And I change the velocity of the activity. And what I find is that when I increase the velocity, the shape of the distribution becomes more Gaussian. For instance, comparing it to the pullback line divergence, with the kinetic energy, if I increase this velocity, this active velocity, the distribution becomes Gaussian, approximately Gaussian. This means that the fluctuations of due to the active noise makes that the work fluctuation much more than the kinetic energy, and as a consequence, we can model the heat without the kinetic energy for the active case. But these are just preliminary results. There's no experimental values here, no real life numbers, just to see the formulas and see the expression. And that's all. Thank you. And I will have to answer any questions.
Pedro?
In order to use this to real systems, which sort of inputs from the experimentalist you need to apply your model?
Yeah, I'm using the IOP model as an approximation. And so I need some bacteria, something that will be explained by the IOP model. And find these values like this velocity, temperature, probably be room temperature. And you also want to see how it's not finished, but by increasing this force, how the kinetic energy also be important or not. But we have some papers we do like next week. Okay.
So what we are seeing is basically that the active term is actually making the importance of the kinetic energy smaller.
Yeah.
Right, because the fluctuations of the active noise, essentially they are larger than the fluctuations from the kinetic term.
Yeah, I have that key, the heat, equal net energy, and having the activity, increasing the activity, makes this have more fluctuations. tonic net energy. So I can use the distribution. That is what the Fulbright version is saying to you.
So as we were discussing yesterday with Lisa, if you confine the particle, probably the relevance of the active term will be smaller, therefore the relevance of the kinetic.
Yeah. Yeah. Yeah.
Yeah.
I think that you need to do the calculation, but probably because the particle will be changing a lot of the inertia.
Yeah.
Since it is confined, right?
More questions? So thank you, Pedro. Okay, so now we have Emanuel Teixeira talking about cell sorting with happy grids. Thank you, Emanuel.
Thank you very much. Organizer, Pablo, Danilo. I will be promise to be short. This is my ongoing project, and I am part with together with Leonardo Pereira and Heitor Fernandes from Universidade Federal do Rio Grande do Sul. So here I actually to present a model. Don't discuss this actually. So we basically introduced model to represent a cell by connecting particles. Cell with particles. We use a kind of particle we call the active particles. These particles can take energy from environment and use it to move by themselves, basically that. So we construct the spring using these particles, and the process which describe the velocities of these particles or these overdeck equations, which has two terms. One term is an active term, which take into account the active particles, and another one term which take into account the interactions between these particles and the energy functions to control to build this ring. So this doesn't matter for now. And to construct this ring, we use a perimeter energy bonds to connect the particles. And we use an area potential to conserve the area of this ring. We are modeling a cell. And we use a repulsion to avoid that the particles cross each other. And the most important, we use a term which takes into account different contraction between different rings. We can mixture and like something like that. So depend on the contact, we can have this tension, and depend on the quantum, we can have another tension. We depend on the kind of the reeds cell. So we mix this, we run simulations with this kind of interaction, and we find segregation process, which is interesting results. And depend on our parameters like activity, shape of the ring, or density. We found a lot of behaviors and segregation process. We use some methods to study this, but for now, that's it. Thank you.
Right on time. Thank you very much. So questions?
I have a question. Which is your starting point? Circular rings, random points?
Now, actually, we connect these rings and they can assume a lot of the formats. Actually, can we use an area potential and we can control how long, which is the format of these rings.
So your conditions are
We
circular or condition?
actually we are using pressure to confine the rings.
Okay, so it's basically like a wall or?
Yeah, like all, yes.
I didn't understand what is the polarity vector?
Sure, perfect. So actually, we define a polarity direction, which the for each ring, where which particle with from these rings follow by themselves. So we have a self-direction propulsion of each rings, and each particle in these rings follow this direction, the same direction.
So each particle of the ring is an active particle, and the ring itself is an active particle as well.
Yeah.
In the sense that.
Yeah, actually, this ring is standard particle, active particle.
So it has an independent direction.
Yeah.
Polarity.
I don't. No, that's
So there's probably like, if I want to think about comparing the effects of the activities, the effects of this different contractility, right? Then there's sort of two-dimensionless parameters I should think about. Like I could make basically a dimensionless parameter out of like the velocities and the damping coefficient for the activity, and then I can make a different dimensionless parameter by multiplying the contractility by some like scale essentially, right? And so in that type, if I were to view that as like kind of a phase diagram of like possible, and it should put them on the same units. Is your intuition based on the simulations you've done so far that the like activity dominates, or is it that the contractility dominates the segregation?
So actually it depends on the ratio between these different energies, because we have an energy term here, and the activity defines some one time scale, different time scale, which it's a little hard to say, because depend on the activity, depend on the interaction between these rings, and we need yet to define the relations exactly to verify that. So
You have a question, the segregation pattern that you see, is it does it depend exclusively on one of the ingredients that you have, or if you turn off contraction or if you turn off the polarity, does it disappear, or everything is necessary for us to occur?
you mean what is necessary to occur the separation? Yeah. In this process, one process is fundamental, which is the rings needed to contract different depend on the kind of the ring, which one ring is touch. That's the reason of this Facebook. It's a little more complicated, but in short words.
Did you just say that lambda 1, 2 needs to be there?
Yeah.
Okay.
That is.
Any more questions? Let's take. So now we have Jorge.
Quem que é?
Na verdade é o Jorge.
Tô...
So Jorge is going to talk about correlated disorder and viscoelasticity of soft colloidal gels.
Thank you so much for this presentation. We are going to present the project where I am working with the Professor Danilo. And we know that in soft matter physics, we can use these periodic lattices in order to extract our solids. But this is only the case for crystalline solids. And in nature, we also have the amorphous solids that they don't have this periodicity in their structure. we have systems that have a disorder. And this is a very interesting family because we actually don't have a generalized theory that explains all of these kind of systems. So we need to focus in the specific features. So, okay? And we are working with subcolloidal gels. In order to describe these kind of systems, sometimes we can use the idea of starting with periodic lattice to introduce a probability of having two different kinds of constants in our periodic lattice. And we can set one of these constants equal to 0. This is some kind of idea of removing links in our system, and this idea we need to be careful with. But this is well using effective medium theories that, for instance, in the current potential approximation, there is a well-known theory in order to, for instance, study self-modes in a rectangular lattice. We will assume that we will have exactly the same constant for all the next nearest neighbors in the lattice, and we can introduce a defect. This defect have a probability associated and of having different values. So, for instance, we can have value equals 0, and we have this idea of removing links. But this is based on the idea that we don't have correlations between the disordered elements of our systems. And in the case of colloidal gels, we actually have correlations between the disordered elements. It means that having a link in a site affects the probability of having links in the neighborhood. So what we are trying to do is to introduce these correlations in our effective medium theory. So our objective is to build an extension of the effective medium theory that takes into account these correlations. What we have seen is that when we introduce these correlations, for instance, the gels become in the sub-isostatic region, it means that it is stable even when we don't have enough links to achieve rigidity. So this is the kind of universal properties we are trying to study with these projects in order to have a better description of our amorphous solids. Thank you so much.
Thank you very much, Jorge. So this is the beginning of the project, right?
Yes, this is the basic idea of what we are trying to do. We are based in past projects. The Professor Danilo have worked with this kind of systems before. So what we are trying to do is to generalize some specific results we have before.
So you want to do a coherent potential approximation?
Yes, introducing the information of the specific information of a fluid that you have, for instance, this correlation. That is part of what we are trying to introduce to generalize our effective medium theory.
And then also compares the simulation.
Yes, that's the idea.
Do you have limits for the lattice size or something like that? I mean, how big can you build this system, or you don't have limits for this?
I think we don't. I think, for instance, when we use this coherent, this effective medium theories, we are assuming that we have the same constant value for all of the lattice. I am not sure about the limit of our lattice, how big it is.
During the CPA, we can check the table and then do an iterate and actually solve it.
Okay.
When we do simulation, of course, then we have to do some short one. The CPA part, the analytic theory.
That's it.
Did you use Newtonian particles or Brownian particles? What kind of particle do you?
Oh, yes. This is at 0 temperature. This is the calculated at 0 temperature. So it's just.
And how do you think the fact that there is now disorder will make the CPA harder? I mean, will the disorder make the CPA analytical derivation much harder?
Correlations.
Sorry, the correlation.
The correlation? Yes, I guess, because well, we had this idea that we can remove these links without taking into account this correlation between we don't care in this effective medium theory, we don't care what we have around, we just say we have a defect here, but we need to take into account something that will take this correlation. So the disorder could be achieved with an effective medium theory, but the fact that we have correlations is the problem we are trying to solve here. So
The lattice can move or not, it's fixed, the lattice. You can found the plug models, for example.
We can apply strain and the plus.
More questions? Let's say.
Thank you so much.
Okay, so now we have our last presentation. Did it.
É, Vera. Tá. Calma aí. Eu não sei por que que não entrou o nome. Esse, né? Eu não sei. É Vera aqui. Ah, tá, Wagner. Tá.
Pra frente.
Essa fotinha.
Yes.
Isso.
Okay, so now we have Vera Henriques talking about porous nanoparticles. Thank you very much, Vera.
Thank you for the invitation. This purpose of this flash talk is to propose that these two light scattering techniques, in the case of nanoparticles, may be aided by some fluid calculations, and we propose a lattice mode treatment. Nanoparticle, I'll focus on this lipid membrane vesicles. Experimental model, main experimental model for membranes are lipid vesicles in solution, and the lipids, as is known, may have I'll show with my hand, charged or neutral head groups. So these objects may be charged or neutral, and the lipid chains have an order-disorder transition, which is displayed through a peak in the specific heat, here 23 degrees. In the case of the neutral membrane, when you have a charged membrane, this transition develops transition in a transition region, which spans here over 10 degrees. And this is also seen in many other through many other techniques, but also through light scattering. And so here we have the neutral scattering as a function of temperature, neutral membrane. So at 23 degrees, more or less, you have also a steep decrease in the scattered light. But for the charged membrane, you will have a region of transparency, which coincides with this region here of the specific heat transition region. And so what is this transparency? I'll focus here on specific measurement, which is using these two techniques for investigation of the radius of the vesicle in solution. So what we show here at that the two techniques coincide in coincident data for the radius, either for the neutral membrane or out for the charged membrane outside the transition region. So you're indicating in red and blue the two techniques, but there's strongly divergent results for the transition region here. So static light scattering, which is a direct measurement of the radius, gives almost 3 times the radius given by the dynamic light scattering. And our explanation is, our hypothesis and explanation is that dynamic light scattering obtains the radius from diffusion. Diffusion is measured. And then the Einstein-Stokes relation is used. But the Stokes relation is true only for hard spheres. This is a result of an analytical calculation. And so if the SLS data are correct, you have a larger radius for the same vesicle. So the only way in which the same volume goes up is through the formation of pores on the vesicle. And if the pores are formed, then you cannot use the Stokes relation. So what we propose that the SLS is correct, but suggests 90% porosity in this case, specific case, from volume conservation. And then we must have an auxiliary fluid, some sort of fluid, some sort of fluid calculation. And what we propose is to use the Boltzmann equation in its discretized version, which is called the lattice Boltzmann, in which you can calculate the drag force viscosity, the drag force coefficient, B, which DLS will measure this, so we can have this from Einstein, which is correct. And so if we do the fluid, I cannot explain the method, but what we obtain is that if, say, you have obtained this 18 drag force coefficient, and this gives a radius of 1 in arbitrary units, the same value of the drag force coefficient can also indicate that you have a sphere of radius 3. This is radius r, this is radius 3r, this is porosity. And coincidentally, at about 90% porosity, you have exactly the same threefold radius here for the same drag coefficient. So our proposal is that you can use these two techniques aided by lattice Boltzmann for the fluid calculations. Lattice Boltzmann code.
Thank you, Vera. Questions? So in principle, you could have a generalized Stokes relation where instead of a sphere, you consider an object of arbitrary shape, but then the problem is that the shape of the object is dynamic.
Shape.
It's changing.
We're really looking at a sphere, but a polar sphere, because it's full of pores. Well, this is not, I should have featured this 2 slides was very difficult. So it's missing the pore. I mean a sphere with pores. So you have, if you have a smaller sphere and with temperature, its radius increases. This can occur if you have pore formation.
So what's occurring is like it's getting swollen due to water or due to solvents?
Yeah, it will get more water inside. Yes, and this would also explain transparency in the light scattering at 90 degrees as I showed, the fact that it swallows.
And through the transition, it does this dynamically.
Yes,
I mean,
yes. These measurements are done in different times of preparation, and so there are equilibrium. So at 15 degrees, this is the radius of the vertical, and at say 20 degrees, it has fallen through the formation of pores, but it does not disintegrate. There are other work which shows that it can remain integral.
This pores, in your opinion, remain steady? Are they forming and closing?
If the radius remains at that size, it's because they are there. They're not closing. But they might be dynamic. Yes, close and open. This data cannot show this for us, I think. Other pores that which can assist.
Any more questions? So let's thank Vera again. Thank you, Vera. So I think this concludes this session. So it took 1 hour as expected, so that's good. Now we have posters and a long break. to chat and have a coffee break.
Vamos falar com o Jorge.